Okay, next, I want to talk about a new application of the chip scanner, and this is the reverse engineering of neuronal circuits, which is more commonly known as connectomics. So the chip scanner can image neuronal tissue, as can any traditional scanning electron microscope. Neurons make up the brain and the spinal cord, and we see several neurons, a bundle of neurons here in this section. The neurons are the fundamental elements of the neuronal circuits. And here we have an image of neuronal tissue that has been stained with osmium tetroxide and also uranyl acetate in order to produce very good myelinated contrast for the myelinated walls here, as well as the neuronal cell sub-ultrastructure can be clearly resolved at very high resolution with this scanning electron microscope. So I would like to tell you now why we would use the chip scanner for imaging neuronal tissue. And so let me please provide some background. In 2013, the United States launched a $12 billion BRAIN initiative. And the BRAIN initiative is to accelerate the development of technologies that will ultimately show how brain cells and complex neural circuits work. The idea is to map the wiring diagram of neuronal circuits in the brain. And the wiring diagram, the schematic diagram of the brain circuit, is what neuroscientists call the connectome. The connectome is completely unknown for humans and most animals. And there is a large effort to explore and understand the connectome and its implications. As for the United States, the European Union has also launched the Human Brain Project, where they, in one sub-program for this brain project, is to use electron and light microscopy to image the structure and determine the function of the human brain. So it's essentially the same goal. And in fact, several countries have launched major new brain research initiatives. Israel, Canada, Australia, New Zealand, Japan, and China have all recently announced major new brain research initiatives. To understand why neuroscientists are on a quest to map the human brain, let me please share with you the story of Anna Bagon, who in 1999 fell while skiing in Norway. Anna was trapped under an icy stream for 80 minutes head first, while her friends did all they could to pull her out. It was 80 minutes had gone by until the medical rescue team arrived, and by that point, Anna was clinically deceased. However, the medical team was able to bring Anna back to life, and this is because her body temperature did not go below a certain threshold. I think it was 14 degrees centigrade. And below such a threshold, cellular damage starts to happen. But the body temperature did not get that low, and so the medical team was able to resuscitate Anna even after 80 minutes of being clinically deceased. Today, Anna lives with all of her memories, all of her skills, emotions, desires, and her senses. These are all generally intact. And it is quite a miracle that a person can be clinically deceased for 80 minutes and then be brought back to life and then have all of these things still intact. Neuroscientists hypothesize that Anna's persona, her entire persona, is stored within the structure of her brain, within the structure, within her connectome. And therefore, neuroscientists are on a quest to answer very fundamental questions. For example, what brain circuitry do we all have in common? What brain circuitry do we all have already present at birth? And what are the associated behaviors? And what are the associated cognitive skills that we have at birth, just based on the structure that we're born with? So let's have a closer look at the neuron. And again, this is all background to explain why the chip scanner is going to be important for neuroscience and connectomics in particular. The neuron spans many length scales. Here we have the cell body. The cell body can be 100 microns in diameter. We also have the dendrites. They can be 1 micron in diameter but span a few hundred microns long. And so here we have quite a range of scales. We would need to resolve dendrites that are 1 micron in diameter, but they're a few hundred microns long, which already exceeds a single field of view for scanning electron microscopy. Looking at other parts, the axon is very interesting. It is also 1 micron in diameter, but it can be 1 meter long, even longer in certain animals like humans. And finally, let's take a closer look at the myelin. The myelin sheath here, there's about 100 sheets of myelin, and there's about a 10 nanometer pitch. So you need to be able to resolve 10 nanometers over scales of 100 microns, a few hundred microns, and perhaps even much longer, up to a meter, in order to really map even a single neuron. And our brains have 80 billion neurons. So this is an immense problem, actually. Interesting about myelin, just that I want to point out, myelin is an electrical insulator, so it's kind of shielding the the neural signal that's traveling through the axon. And the degeneration of the myelin sheath is a hallmark of several neurological disorders, such as multiple sclerosis. So, the neuron spans many length scales. There are many neurons to map. And furthermore, the neurons are all intertwined and forming synapses with each other in 3 dimensions. And therefore, electron microscopy with 3D stitching is very important for mapping neuronal circuits. And several groups around the world are already off and imaging small sections of neuronal circuits. Here is one sample preparation method where an in situ ultramicrotome is used to slice neuronal tissue right underneath the scanning electron microscope. And between each slice, a new scanning electron microscope image is captured. And the idea is to stitch these over volumes that are over dimensions that are about 1 millimeter or even up to 10 millimeters is what neuroscientists desire for capturing the essential structure of neural circuits. Now, this will not be an approach for mapping the connectome of the entire human brain. Certainly, this is a very slow process, but still, the ability to do this at the scale of 1 millimeter and 10 millimeters is very important already, can show a very important information for neuroscientists. So there are challenges with the electron microscopy, and these challenges are starting to become known in the neuroscience community, as illustrated by this 2015 paper. Here in this figure, you see that large overlaps are being applied and software is being used to try to correct for stitching errors in the traditional electron microscope. Now, this is essentially the same challenges that semiconductor reverse engineers have been faced with over the last 15 years. So neuroscientists and semiconductor reverse engineers essentially have the same problem, and that's reverse engineering of circuitry, and the chip scanner is therefore, with its excellent 3D stitching, the chip scanner can really enable neuroscience in addition to semiconductor reverse engineering. And so together with our partners at the George Washington University, we set out to make a map of a mouse spinal cord using the chip scanner. And so here we used 100 micron fields of view that already had corrections built in, and the laser interferometer stage of the chip scanner, and we stitched together many such images of the mouse spinal cord. So here you see the axon cross sections, these circles. They're myelinized, so you can see, even resolve the myelin sheets in this image. And you can even see some of the stitching boundaries. Again, these are not displacements in X and Y. These stitching boundaries are due to changes in signal intensity, which is another dimension of stitching errors. And signal intensity changes, that can occur quite easily if there's damage happening to the sample or charging. This can change the signal intensity, and therefore it's quite common to find these boundaries of signal intensity in these stitched images. Okay, so zooming out now by a factor of 3. So we have recorded even a larger area of this mouse spinal cord. And finally, we can zoom out by another factor of 2. And here we see that we have, in fact, imaged the entire cross-section of the width of the mouse spinal cord. So it is important here to note that this is the most accurate large area, high resolution map of neuronal tissue directly acquired by a scanning electron microscope instrument. This image contains about 5 times 10 to the 10 pixels, and each of those pixels have locations that are known to the accuracy afforded by laser interferometry. We therefore believe that the chip scanner has great potential as an enabler for connectomics. So as for semiconductor reverse engineering, it is important to have a variety of electron detectors for imaging of neuronal tissue. And here we show just 3 different images of the same small area of neuronal tissue. Each image shows different information. This is a secondary electron image that shows topographic information. Here's the energy selective backscatter detector, in-lens detector that was used to show a very high resolution but slightly noisier image. And finally, here's the post-lens angular selective backscatter detector as well. So a variety of imaging conditions is possible. And this is important because there are a variety of ways of doing sample preparation. It is quite an art. Quite a lot of effort has gone into properly preparing these samples to fixate these cells with osmium tetroxide and further with urinal acetate and preserve the ultrastructure and the myelin sheets. It is quite an art in itself. So there are a variety of ways of preparing these samples. And so it's important to have flexible instrument detection capabilities. Here's another sectioning sample preparation method. This is the automated tape collecting ultramicrotome method. And this is interesting because the brain sections are continually sliced and collected from the water bath here, and they travel up along this tape, and these tapes are then laid out down onto silicon wafers. And then these wafers go into an electron microscope for imaging. However, the wafers, they're not perfectly flat, and so keeping the sample in focus is certainly a challenge for this method. And so we believe that the chip scanner would be very well suited for this method because it has a height sensing apparatus that is used for keeping the sample in focus over large areas.